All right. Hello, everyone, and thank you for continuing to join us throughout the day here at the Lytham Partners Spring 2024 Investor Conference. My name is Robert Blum, Managing Partner of Lytham Partners. During this presentation, we welcome Shuttle Pharmaceuticals. Shuttle is traded on the NASDAQ under the symbol SHPH. And joining us today from the company is Dr. Anatoly Druchilo, the company's chairman and CEO. Before I do turn it over to Dr. Druchilo, I just want to remind everyone that management is available for one-on-one meetings throughout the day here today. If you've not already signed up for a one-on-one meeting and would like to do so, you can send me an email. Email address is Blum, B-L-U-M, @lythampartners.com, or you can visit the conference landing page, lythampartners.com/spring 2024. From there, you can click on the investor and attendee registration tab and make your one-on-one selection. So with that, Dr. Druchilo, the floor is all yours.
Hey, good afternoon. Thank you, Robert, for the introduction. Good afternoon, investors. And what I'd like to do today is to provide you with an update, basically a status of the company at this point in time. So you can see that we've managed to reach several of the milestones, and we're ready to initiate the clinical trial and to initiate some preclinical work. So Shuttle Pharma, this is the forward-looking statements, the disclaimer. Please read this at your leisure. And I'd like to address again, for those of you who may be new to this, the fundamental problem that we're trying to address. The ability to deliver high dose of radiation to control cancers is outstanding at this time. The technology in terms of the machines, the linear accelerators, the proton therapies, the cyberknives, all of these pieces of equipment are very, very, let's say, controlled by computers, and they're able to target tumors anywhere in the body, and we can deliver any dose we want. However, in treating cancers in human beings, our problem really persists, and that is that the tumors are frequently either adjacent to a very sensitive normal tissue, or it's incorporated within a normal tissue. So that our ability to give a dose that we want to give is usually less, and we are able to then, let's say, end up with a suboptimal dose to the tumor and the resistance to radiation and recurrence of cancers. So this figure shows you a couple of pieces of equipment that we utilize. The CT scan through the chest shows where we're treating a tumor of a lung, and then we can see that there are normal lung and heart and other organs that are treated to high dose of radiation. And that would be limiting. You cannot go beyond what the heart and the lung and the vascular system can tolerate. And the same thing over here on the right with the brain. This particular treatment was using proton beam, and as you can see, we can shape it very, very nicely, but then you still have an intrinsic problem of normal tissue in between that you do not want to damage. So one of the ways to deal with that is to sensitize the cancer cells to the radiation therapy. This diagram, the curves show you that by adding a sensitizer, you can move the tumor control, the tumor control effect to the right to a lower dose. So that in effect, if you can have to stop at a certain dose of radiation to a location, by adding the sensitizer, you make the tumor more sensitive, and if in effect, you're giving the equivalent of more radiation. So that is one of the ways that we want to deal with this. There are a number of drugs that are used off-label, and our drug would be something that we want to develop. It's called ropidoxuridine, and this drug is capable of sensitizing cancers directly by being incorporated into DNA and making the DNA more sensitive to free radicals. The other way to approach this is the current approach is with activation of the innate immune system. We know that in some cases, when radiation is given, that the immune system is activated in patients, and even tumors that are outside of the area that's irradiated become affected, and we see responses. This is something called the abscopal effect. It doesn't happen, to a great degree very often, but when it does, it's a remarkable observation. Now, many of the efforts now with the agents that stimulate the immune system have been added to conventional radiation in an effort to improve the outcomes for these patients. And here I show you that there are at least 2 studies using these monoclonal antibodies that have been used to improve the responses for the treatment of non-small cell lung cancer after radiation. We have a drug called HDAC6 selective inhibitor, which enhances the immune system and has been observed in preclinical models. The company overview, for your purposes, has 2 divisions. We are the holding company, Shuttle Pharmaceuticals Holdings Incorporated. And then within that, we have Shuttle Pharmaceuticals Incorporated, which is the drug therapy division. And then, of course, we have Shuttle Diagnostics Incorporated. That is the diagnostic division. And I say divisions, but they're both incorporated as separate corporations. This is important for you because this allows us to take some of the intellectual property that we have developed over the last 2 years and develop them with another, let's say, focus, the diagnostics and theranostic focus. The therapeutic division, our main drugs, our principal ones are ropidoxyuridine, which is an orally administered radiation sensitizer, which means that the drug can be taken by mouth as a pill every day at the time of radiation and can then help to increase the sensitivity of tumors. The other is the HDAC6 inhibitor. Our drug is SP2-225. And what this does is after radiation, there are many antigens that are activated in these cancer cells. And some cancer cells that are still able to recover have these antigens on the surface, and then the innate immune system can attack them and help prolong the ability to control the tumors after radiation. The other 2 areas that we have focused on, and this is the diagnostics, we have developed something called the PC-RAD test, which is a blood test for predicting outcomes of patients who are undergoing radiation therapy for prostate cancer. The idea being that if you have a favorable finding on this test prior to treatment, your chances of having a good result are increased. And whereas if you have a negative finding, your chances are decreased, and perhaps you would benefit from some more aggressive therapy that may include things like chemotherapy or more aggressive radiation. The second one is something we call PSMA-B ligand. This is a molecule that's a theranostic and was developed to both use for diagnosis and for therapy. We developed this to make the proton radiation therapy a little more effective. So what happens here is that the PSMA enzyme is located on the outside of cancer cells, prostate cancer cells in particular, and the ligand binds to this PSMA. The PSMA then gets incorporated into the cell, and we're able to deliver treatment more effectively by targeting those cells. This can also be diagnostic because if a small radioactive imaging tracer is attached to the PSMA-B ligand, you can then do PET scanning to monitor the location of any disease that has spread in the patient. Okay, so this is an overview of where we are in terms of these 3 areas. The sensitization, ropidoxyuridine is our lead for that, and we're getting ready for a phase 2 clinical trial. The immune modulation, HDAC6 is a preclinical a molecule, and we are working to determine the parameters that we can study in preclinical models, basically in mice that have intact immune systems. We're collaborating with scientists at Georgetown University for that. Then we have the diagnostics and theranostics. The PC-RAD test was developed. I was one of the inventors of this, and this was developed at Georgetown University. We have licensed that product for the company. And then we have the PSMA-B ligand, which we have basically invented as a new molecule with the help of chemists that have collaborated with us. Okay, to give you a little idea of how do these things work, I have a couple of diagrams which may be helpful. So the drug that we are interested in is something called iododeoxyuridine, and this is this drug right here, and it's it's a type of thymidine analog which would be incorporated into the DNA of actively growing cells. This material, when it's incorporated into the DNA, it helps to develop, when radiation is applied, additional reactive free radicals. And those free radicals break DNA, and you end up with a greater proportion happening in cancer cells because they're actively growing, as opposed to normal cells, which are more in rest phase. So you have a proportion of about 2 to 3 improvement in the uptake in growing cells versus resting cells. Now, our drug is ropidoxuridine, which is IPDR, and it is metabolized after taking it in the liver and in tumors to become IUDR. And this is important because when IUDR is given, it has a very short half-life in the bloodstream. And the tests that were done 25 years ago, there were some studies that were performed by the National Cancer Institute and University of Michigan, and reported in the literature, showed that in astrocytomas, you saw about a year improvement in survival. And in the glioblastomas, which are the very aggressive brain tumors, you saw about 6 months improvement in survival when you added the drug to the radiation. The problem with this drug was because of a very short half-life, you had to have a continuous infusion into the patient's bloodstream. Now, this added to complications and technically was difficult. And what would happen is that normally you can give an infusion, it's not a problem. But if you have to do it for 6 weeks, it becomes a problem, because it's continuous 6 weeks. So that the IUDR is delivered in our case by the prodrug, which is metabolized to IUDR, and the IPDR is the drug ropidoxuridine. Now, we have received money from the National Cancer Institute to do the phase 1 and pharmacology study. And we've achieved a dose of 1,200 milligrams daily as the maximum tolerated dose. The patients these are very advanced patients, but they showed a distribution of stable disease and partial responses as shown here. And in the phase 2 clinical trial, we want to test this in the most aggressive brain tumor that we could find. And that is something called IDH wild-type methylation negative glioblastoma. This particular group of patients at this time really has only radiation as the standard of care, and more than half the patients do not live beyond 12 months. Okay, so that we have a very, very focused, targeted patients, and they're because we picked the most aggressive, we think that this is an opportunity to either test the validity of this hypothesis that this will work better or not. So we do have a yes or no type of study put together. The IND was approved by the FDA, and we received a safe to proceed letter. We received guidance from the FDA to use 2 different doses. The 1,200, which is a maximum tolerated dose, and 960 milligrams per day, which is a 20% lower dose. And that's because the general idea of the FDA is that many of these drugs sometimes can be given at less than the maximum tolerated dose, and perhaps then there would be fewer side effects So we will do this randomization to determine which dose is optimal. Then the optimal dose, and let's assume that's 1,200 milligrams. An additional 14 patients would be added, and that would give us the statistical significance to determine, based on a survival endpoint, whether the drug compares favorably to historical controls or not. This trial has been prepared. We've engaged Theradex. They have prepared all the necessary paperwork. The clinical trial was approved by a central IRB and is now going through secondary approvals at each of the institutions. We've identified 6 academic cancer centers to do the clinical trial. And we feel that with that many centers enrolling, that we would be able to complete this part easily within 12 months. And then this would be another maybe 6 months. So we're talking about 18 months before we get some sort of 18 to 24 months for a readout of the endpoint. Now, the preclinical work, the immune modulation, we focused on SP225. I should say we have a large number of HDAC inhibitors that are discovered and owned by Shuttle Pharmaceuticals. We have no royalty payments owed to anyone. And what we're doing is to develop the ones that we've selected. There are 2 that we've selected, one for treatment just in general, immune therapy for various diseases, and another one for the treatment of breast cancer, ER-positive breast cancer. And as you can see here, in this preclinical work, we saw a delay in the regrowth of tumor by combining the vehicle with, by treating the tumor with vehicle, stimulating the immune system, and basically in the cartoons that are provided there, I give you the reference. We've published this work in collaboration with the Georgetown University scientists, and the next step would be to irradiate cancers and then do it fractionated, large fraction, and then basically do the IND enabling and go to a phase 1 clinical trial. Our limitation for this will be the rate at which we have resources available, because of course the clinical trial takes precedence, and we're putting most of our resources toward getting that to the next scientific milestone. So the intellectual property that I mentioned, ropiduxuridine is being developed under orphan disease designation. We received approval for that use of that drug for glioblastoma. And then, of course, we have developed the next generation, which is a combination with something called Tiparasil, which allows for longer lifetime in the bloodstream. So this helps to prolong the bioavailability of the drug. The HDAC inhibitors, we have the 2 that we're interested in particular. We have approximately 20 plus patents on these various drugs. And then these 2, the 225 is for the immune response, and 1303 is for the breast cancer indication. The 2 tests that we talked about for diagnostics and theranostics, the PC-RAD test, this is a kind of a schematic showing you. It's basically a kit that involves a number of reagents and controls. And then we use mass spectrometry to test the plasma from patients that are undergoing radiation. We do this before they decide whether they want radiation or surgery. So, for example, if a patient had a negative, let's say, result from this, that may be an indication that the patient should maybe have surgery instead of radiation. So there are these types of considerations. The test itself is something that will need, we've performed the discovery, we've performed the analytical validation, and this time we're looking for the clinical validation. The funding that we'll be seeking will be for doing a clinical trial where we collect blood from patients and then just follow them. We have several cancer centers interested in participating in this. The last one is the PSMA-B. The PSMA, as I mentioned, is an enzyme that's also an antigen on the surface of prostate cancer cells and prostate cells in general. The PSMA ligand has been developed over the last 25 years. Novartis is using one version of this ligand to do diagnostics and therapeutics with radiation for personalized medicine, and that is a billion-dollar drug for them. We have some other indications that we're interested in, but certainly the diagnosis and therapeutics is also a possibility that we will be exploring. We'll be looking for collaborators to work with us on this particular intellectual property. So our priorities as they stand now are, the first priority is obviously to develop the ropidoxyuridine and RT for brain tumors. Okay, and if the completion of the phase 2 that we're planning to do would put us in position to seek breakthrough designation and to also prepare a randomized phase 3 clinical trial, and that would be something we would do with FDA guidance, and with that we would look for full marketing approval for the indication of glioblastoma. The second therapy is the HDAC6 selective inhibitor. We need to do you need to manufacture some drug and to perform the IND enabling studies so we can get that into the clinic at the phase 1 level. The diagnostic priorities, the predictive biomarkers for prostate cancer is a fairly straightforward blood collection protocol, but it does require 360 patients. So we will have to seek additional funding to advance that. The institutions have been identified for this, but we will be seeking additional funding over the next 6 months. Diagnostic priority 2, PSMA ligand for what's called theranosis, which is for the diagnosis and for therapy with the drug, and it's involved with personalized medicine, specifically for prostate cancer, specifically for patients with metastatic prostate cancer. And this is an intellectual property that we will be seeking joint venture or collaborations with other institutions or other companies. Our financial overview at this time, we have it's about around 17,000 shares out there. We have a market cap of 7 million, and we have a cash balance at this point in time is sufficient for us to go out and raise additional capital. The Shuttle's team, leadership team, is experienced. I have worked with, in fact, Dr. Tyven Rich and I know each other for over 40 years, and we've worked together on other things. We went to school at Harvard at the same time. He went to Mass General. I went to the Joint Center for Radiation Therapy. My son is a hospital administrator by training, and he is a participant in managing our day-to-day operations and our personnel. Dr. Mira Young is a molecular biologist, a scientist. She's helped to discover and test some of our intellectual property. Scott Grunrod is a medicinal chemist, PhD medicinal chemist, and he actually synthesized many of these drugs and is on all of our patents. And Mike Vanderhoek, he served as the administrative director of the Lombardi Comprehensive Cancer Center prior to joining us, and he functions as a CFO and vice president for regulatory affairs. We have an external board of directors that includes Steve Richards. He's a CPA and he's involved in media, making movies, but he basically, I'm sorry, something happened here. But he basically provides us with the necessary backup for our audit team. Milton Brown, MD, PhD, Vice Dean for Research, East Virginia Medical School. Betty Jacobs, Professor, Distinguished Scholar at Georgetown. She was the former Dean of Nursing at Georgetown University. Chris Seniaki has held big pharma positions, and is the founder and chief and the CEO of TCG Green Chem. And Joshua Schaefer, as an MBA, he's a commercial officer, and he basically has had also big pharma experience. That's our team. This is our story at this point in time. We're looking to initiate the clinical trial as soon as the contract, the first contract is signed and the approval of the IRB is provided to us. So we're looking at probably July or early August as our time frame. And of course, the problem with these things is always we're waiting for someone to sign off on something, but we have 6 centers, and I think we're looking at something coming very soon. And I look forward to making that announcement to you as soon as it happens. Thank you very much.
Fantastic presentation there, Dr. Chillo. I think you sort of touched on the key point there, which is for investors to understand really the key sort of milestone or event that they should be looking for as it relates to the upcoming phase 2 trial initiation. It's as you just mentioned there, waiting on sort of centers to sign up. Anything else you can sort of expand upon there?
So the key to this company is can we show sensitization and prolongation of survival in brain tumors? The fact that we have picked the most aggressive brain tumor, the one that the patients do not live long at all, and for which there really is no standard of care outside of radiation. The temozolomide that's used in glioblastoma does not really work in this group of patients. So we really do have something where I feel comfortable that this is a disease that is worth going after. And even though it's, let's say we don't get a home run with cures, but we do get prolongation of survival, I think that would be a good indicator of efficacy of this as a sensitizer. And this particular sensitizer could be used in other disease sites, including things like lung cancer, head and neck cancer, and GI cancer. So there are other potential indications, but obviously the first indication is the important one, and I think we have a good focus. For the other drugs that we have that are preclinical, we have, as I said, we have 2 HDAC inhibitors that we're working on, and the idea is that the animal work is something that will direct how we go forward. But of course, the safety and testing and IND enabling will determine which ones we put our resources behind to develop. The SP2-225 is the next one I'd like to do. The next milestone would be manufacturing the drug in a larger quantity to scale it up, and that's going to require the next phase at the additional capital raise to really take it forward. We can manufacture it, but then, for example, to do the IND enabling typically costs about $2 million to perform and then once you do that, then you can take it into the clinic. So those are the milestones and the barriers that we have to overcome, which I feel very comfortable about being able to do it. I think we've come a long way. We've manufactured drug in a pretty efficient way for the ropiduxuridine for the phase 2 clinical trial. And we have a good team, including good CROs that we have experience with now.
Excellent. Very good. Well, we'll go ahead and leave it there. Thank you again so much for the presentation. Really informative. Want to remind everyone, if you'd like to schedule a one-on-one meeting with Dr. Chillo here and the team, shoot me an email, Blum, BLUM, at lythampartners.com, or again, visit the website, lythampartners.com/spring 2024. Click on the investor and attendee registration tab, make your one-on-one selection. We'll go ahead and get you started. So again, I hope everyone enjoys the conference. Thank you again, Dr. Chillo.
Thank you very much.